Carrillo. José Antonio Carrillo is professor at the University of Oxford, formerly at Imperial College, at the Autonomous University of Barcelona previously, and at the University of Granada, where he wrote his thesis. He is a highly recognized specialist in partial differential equation, in particular in kinetic theory, and currently he is vice president of the Society of for Mathematical Biology. It is a real pleasure to have you with us again. Today's talk is about aggregation-diffusion models for differential adhesion and application. Thank you to accept this invitation.
Thank you very much, Juan and David. It's a great pleasure, as you know. I mean, it's always very good to have to be in my alma mater university, even if this time is only virtually. I'm always happy to visit, as you know. And today, I would like to concentrate in several aspects of the use of aggregation diffusion models in mathematical biology, but I want to concentrate mainly on this aspect of the differential addition. So I will showcase these applications with two particular experiments. In fact, this experiment that we see, these snapshots here now in the slide, is the experiment that triggered my attention to this from the very beginning, like that was like 6-7 years ago. And a collaborator of mine now, Hideki Murakawa, showed me these experiments that he was trying to model with a biologist, Hideru Togashi, in Japan. And here what you see is a cell colony that has been grown in lab. And in the first row, what you see is these cells just growing, and but they haven't been. They are the same kind of cell, even if you have this different color. This is just artificially for the experiment, just to make a distinction between the two sides of the colony, say. But they are essentially exactly the same type of cell. They are some hepatic cells from mice. It doesn't really matter the type of cell at this point. And the main point here, from the biological point of view, is that they can control the leveling at which certain proteins are produced in the cells. So they can essentially control, in this case, certain nectins and another type of protein and cherry, that they control somehow the ability of these cells to adhere to for adhesive bonds with nearby cells. So in the first row, they didn't do anything. They just let them grow. And what you see in this experiment is when they meet in a very nice front. While in the second one, they were able to change that ability, as I said, by changing the level in which these proteins are expressed. And then they produce some mixing zone. So this was somehow one of the experiments that triggered my attention. And then also another one, the second one, that I will come back later on in my talk, is about pattern formation in zebrafish. This is a model organism that have been used for pattern formation for many years, and there are several groups in the world that have been look at these kind of issues, especially the lab around Shigeru Kondo in Osaka University, and Musla in Bollard in Max Planck Institute in Germany, and also a group in Maryland that I now forgot the main driving force there. But what essentially the question here is how these stripe patterns form in the skin of zebrafish, growing zebrafish, based on the interaction of very few type of cells. In fact, it's very well known what are the cells that are involved, and very detailed properties about the interaction of the different type of cells. I will come back to that later. So these are the two experiments that trigger my attention to this. And let me start from very basic models. So just to explain the basic ideas. So the very basic model I want to discuss is, let's assume that we are working with a group of cells in which I'm trying to model cells in the easiest possible way. So since I want to model a very large population of them, so I don't want to enter into any kind of detail about the shape of them, but just rather I model the center of the cell, the center of the nuclei, if you want, of the cell, and that is what is the position of those cells. So I can think about the cells as point particles. So let's start from the very beginning with this very simple way of thinking. And I'm going to model the interaction of the different type of cells by an effective force that takes into account the different interaction with nearby cells. So how I model this, I mean, this is just to have an idea of what I want to do. So I give myself an interaction potential that I call W, and then I look at the forces that one particular cell, particular particle here in the ensemble receives from all the others. So gradient Wxi minus xj is the force that the kind of particle xj is producing onto xi, and here I have the average of them. Why I have the 1 over n here? Because I'm assuming that this is part of the assumption in the modeling, and when I increase the number of cells here, I want to keep the force of order 1. So in the sense that if I assume that the forces that are produced between the particles are of order 1, the average force I want to assume that is felt by one of the cells here is also of order 1, even if n is large. So, and also I'm doing a kind of a relaxation limit already, so I neglected inertia terms, so that's why I have a first order system. So what's the point of view here for the modeling? I want to coarse grain this kind of very simple system to a PDE. How I do that, this is what I'm going to say is very classical now, is what is called the mean field limit. So what you do associated to the interacting particle system here is you define what is called the empirical measure, which is a sum of Diracks located at the solutions of the ODE system with the right normalization. And you look at the PDE satisfied by the empirical measure. And in this particular case, it's easy to do if W has the right assumptions. But in general, the question is what happens when the number of particles is large? What is the PDE that you can get? In this easiest sample, in the easiest sample in which W is smooth enough, the PDE that you get is of this form. So what you get is a continuity equation where the velocity field is a mean field in the sense that is minus the gradient of the potential convolved with the density. So what you get is a continuity equation with a nonlinear velocity field depending on the density itself. The interesting thing about this particular problem and the technicalities involved is that depending on the W, you can get well, many different properties of the possible, for instance, stationary states of this system. That's not too important now for what I want to discuss, because what this is just an idea to give you an overall strategy of what I want to do in the case of having more than one cell involved. And also to make it more realistic, we are going to do all the modeling based on the potentials. So this has been done already with different perspective, but similar ideas in the literature. So here I'm mentioning some of the people involved on that, Gerish, Chaplain, Sherratt, Murakawa and Togashi, and also some of my collaborators before, Colombi, Shana, and Luigi Preziosi. So The idea is to incorporate at the level of the potential some of the more realistic assumptions that you can use these for cell interactions. So here, what the assumption I'm gonna do is that the potential is very strongly repulsive at a certain, if the distance between the cells is smaller than certain value. And this value usually, I'm gonna take it as twice the typical size of the nuclei of the cell. So essentially, what I want to model by this repulsion is that I have a volume constraint in the system, that I cannot compress the cells too much because they are simply, they have some cytoplasm inside, and just by mechanical properties, you cannot compress them more than certain distance. So I want to introduce that in the model by putting a very strong repulsion for short distances. The adhesive forces, the ability that they want to attract and to produce bonds with the nearby cells, is modeled at a middle range, and mainly because the cells are producing this, and many of the cells involved in some of the experiments that I showed you before, by producing long philopodia or large protrusions, and when they touch, they produce certain bonds, and they adhere to each other, or they in fact repel each other in some sense. They don't produce the bond. So I will model this kind of adhesive forces in a middle range, and finally, if the two cells are at a certain distance, I will not include any interaction. So there will be a limit of so the potential in some sense is completely supported from that point of view. Okay, and also if you want to do that for two species, like I had in the experiment of Togashi about cell adhesion, then I will incorporate these by, and I will incorporate the fact that the strengths of attraction could be different for different type of cells by assuming different potentials for the different interactions. So at the level of the system of two particles interaction, you could write a system of that kind, again with certain number of particles for each of the species that you can write in the mean field limit, again, if you want, as a system now of interaction or aggregation type equations, in which you have the velocity field felt by one of the type of cells is a sum of the forces produced by the two densities with possibly different interaction potentials. Okay, so let's assume, I mean, I'm gonna simplify a bit the notation. And here I was talking about measures because that's the way in which you were producing the mean field limit and how you get to these equations. But now I'm gonna, in fact, do two assumptions. I'm gonna simplify more the model. So the first assumption I'm gonna take is what I mentioned before about being the potentials very strongly repulsive at zero. How I'm gonna model this? In fact, I'm gonna take it to the stream. What I'm gonna assume is that the potential is some nice potential that will model either attraction or repulsion, depending on what we are interested in, plus a Dirac at zero. And this Dirac at zero is going to be the part of the strong repulsion. And if you do that, if you just formally put a Dirac here, what you recover, I mean, just do it here, you put the Dirac here, of course, the gradient, you will put it on the density. So what you will get is density times the gradient of the density. So what you are doing is extracting from each of the four potentials terms of the form epsilon, gradient rho, epsilon, gradient eta, and so on and so forth. So at the end, what you get is this additional term here. And if you think now about this final system, what you have is this part that acts as kind of the mechanical part, in which what is happening, you can interpret this now as that each of the density is feeling the pressure of the total population, because rho plus eta is the total population, and the gradient of rho plus eta minus gradient of rho plus eta, because I'm writing the right-hand side, is the total pressure produced by the total population. So in fact, that's the way of interpreting this. And that's the term again that is going to give me the volume filling or the volume constraint feature in the model. So I would refer to this kind of system as an aggregation diffusion system. And now let me look at it from the point of view of an applied mathematician. So I'm going to try to understand what is the phenomena, or the most interesting phenomena I can get out of these systems. So let's start with the one-dimensional case, and let's see what do we learn on the one-dimensional case in terms of, for instance, stationary states. And to concentrate the discussion, let's look at two particular cases that are important. I'm going to take now W11 and W22, so the interactions between the same type of cells as equal, and I'm going to take them equally to just x squared in one dimension. So they are just fully attractive. They like each other with certain very much, with a certain very strong repulsion. While the W12 and W21, I'm going to look at two particular cases. One in which they attract each other, so W12, so the species, the red species likes the green one, and vice versa. While the second one is going to be attractive repulsive. The red likes the green, but the green doesn't like the red. So the love is not bidirectional here. Okay, so let's look at first the case of attractive. And as I said, I'm going to take particular cases. So I'm going to take the x squared for the interaction between the same species, and I'm going to take modulus of x in one dimension and the interaction between the two different species. And why do modulus of x? Well, I just take modulus of x because in this particular case, modulus of x is the fundamental solution of the minus Laplacian in one dimension, and that simplifies certain computations. So steady states of the equation, if we go back here, we can see that if this quantity that I have inside the derivative is constant on the support of rho, that is going to be a steady state. And the same here, if this quantity is constant on the support of eta, this is going to be a steady state. So let's look for particular solutions that are steady, that they satisfy precisely these conditions. Let's see if we can find them in this particular case of these particular potentials. So in fact, if you work with that, it's not difficult in this case to see that you can find explicitly, or you can write explicitly what rho and eta are going to be in each of the components of the support. So, whenever one of them is present and the other is not. This is what this means. So they have to be a combination of x and x squared, and when both are present, they have to be combinations of these sines and cosines with certain constants. Here, there are constants that we know, which is little m1 and little m2, which is the mass of the population rho and the mass of the population eta. So one particular solutions that you get is this kind of solution. So this kind of solution in which one of the species, let's say the red, gets completely engulfed by the blue. And in fact, you see that it has this very sharp interface at the boundary of the support in the red. And in fact, it's really sharp. What you have is a jump here, okay? So you have a jump discontinuity at the boundary. So the interesting thing about this is that you can get precisely steady states of this type in which you have one species engulfing the other completely, and with these sharp interfaces. Let me show you now some. Let me see if how this works. Some simulation about the steady state. I mean, here is the initial data is just constant in certain interval, and you will see now the evolution. I'm not discussing here that much in the numerical simulation, but if somebody is interested in the numerical schema inducing, can ask me later. What you see, the interesting feature here is that you develop these kind of sharp interfaces as t goes to infinity. And here you have another example in which initially they are not that mixed, and then finally they get into that steady configuration. And this is just for different initial data and other potential. Okay, so let me continue with the case. Well, you can compute them even explicitly in this case, as I said. And the basic to compute them explicitly is that in this particular case that we call the Batman profile for obvious reasons. This Batman profile, you can compute it by just putting the constants on the masses, assuming that the total population is continuous everywhere, in particular at the point at the end of the support, assuming that you have a support of rho larger than the support of eta, and then you can compute all the constants and find it explicitly and see that you have a unique solution of all for the constants that are involved. Okay. So that's the evolution I was showing you before. And let's go to the case of attractive repulsive interactions. When we have that one of them is attracting, in that case, the interesting feature that happens is that you can get completely segregated stationary states that, like here, or segregated states but that are adjacent. So at the boundary, you have these sharp boundaries, these sharp interfaces. So again, some of them you can compute them explicitly if, in this particular case. And depending also on the parameters and the choices of the initial data, what you can get is traveling pulse solutions, traveling waves, instead of stationary states. But again, a video explains this probably much better than words. So let me go to the next. So here you have the simulation of in this case of attractive repulsive. You see that even if at the beginning there is an overlap in the supports of the two species, as t goes to infinity, they get segregated. The little intersection that you may have here is just due to numerical resolution. If you just improve your resolution, you will see that you have a sharp interface. And here you have some kind of traveling wave instead, because the initial data, the distribution of mass, is not symmetric around the center. Okay. So what does it choose exactly that you're going to have segregation or not, and that you have this possibility of overlapping or not of the two species? So this is one of the interesting questions at the theoretical level. Let me just mention that in fact this is already present even in the simplest case, when you don't have any interaction. So when you don't have any interaction, I mean no local interaction, when you have only the population pressure, this very simple model, if you have a very easy way of solving this model, which is to add them. If you add them, you have an equation for the total population, which is nothing else than the porous medium equation, nonlinear diffusion equation, with exponent 2. So in fact, this question is even present at the level of the porous medium equation. It's just looking at it from different perspectives. So here I'm solving this equation with an initial data which is separated, okay, segregated. Rho is on the side, eta is on the other side. You see that they never merge. And of course, the sum follows the solution of the porous medium equation, okay, with exponent 2. This is a typical profile of a Barenblatt. Now at the boundaries, because I have no flux, at the end, everything goes to constant. Here is just the same with some mixing. So, and then you see the self-similar profile of the Barenblatt with the mixing of the two. But you see that this behavior is even already present in the very basic model without any interaction. Okay, so in fact, we look at the system with general, with general population pressure instead of just given by the sum with any general nonlinearity here, and with also reaction terms. I'm not going to discuss too much about the mathematical analysis of this system, but we were able to analyze this well, at least in one dimension. There are several other results that I will mention later on in several dimensions. But again, what we were able to do is the case of the reaction terms. When you have these drift terms, these non-local terms, still we are lacking a good system theory, a good well-Posner's theory for the system because of the formation of these sharp fronts. I will come back to that later if I have time. Okay, so just to show you again some simulation in the case of, here is the case of with reaction terms. So we have some kind of logistic growth in the right-hand side. So the two densities are not conserved, but each of them, they grow for a while. But you see when they meet, they produce a sharp front if they are initially segregated. And this is what we prove in the case of the reaction-diffusion system. In the case of the reaction-diffusion system, we prove if they initially are segregated, they remain segregated. This is another example on the evolution in time. Okay, now that we have learned some bits on one dimension and we get some ideas of what are the interesting questions, the difficulties, can we go further? What happens in 2 dimensions with this? So in fact, that's what we, yeah, that's probably the most interesting from the application point of view in mathematical biology, the most interesting question. And here I'm going to show you simulations of the trying to mimic the experiment by Togashi with 2 types of cells. This is the initial data for the system. I will explain you the system that we use here in the next slide. Here, by just playing with one parameter, which is the ability of adhesion between the 2 types of cells, the attraction force, the strength of the attraction force between the 2, you can go from this case in which, from this case in which what you get in your evolution is segregation, full segregation, to by changing the parameter to this kind of stationary state in which you have a sharp interface, and then you have again the sharp interface in which one of them is engulfing the other, and finally you can get even to the case of just mixing. The case of mixing is when they are the same type of cell, essentially, because the same attraction between the 2, and you can write an equation for the sum. So as soon as you make it asymmetric, you can get all this kind of behavior, which was well documented in what is called cell sorting. So this is what, okay, let me see. This is how we built the model. So to make it more realistic, I mean, just summarizing, what we have is for each of the populations, sorry about the change of notation, here is just one population is called U, and the velocity field of the continuity equation is obtained with one part, which comes from the total population pressure, that is going to be, in this case, gradient U. And the other one is done non-locally with some kernel. Okay, and you can write the non-local way explicitly the convolution for a radial interaction. And also we put a term here, 1 minus U, that gives me a saturation on the forces to make more realistic the behavior of cells that were immersed on the other type of cell, that they will feel at middle distances a force in the direction of the highest concentration of the other type of cell, which is what is a behavior that is considered more biologically reasonable. Okay, so here you have a simulation of that kind of just one equation again, and you see the formation of these local droplets, kind of concentration of the density. And also you see that it can change very slowly in time. And some of them, they will merge eventually until arriving to a steady state. Still they are merging here, okay? Until they arrive to a steady state. Okay, so that's the model that we used finally for the experiment of cell sorting by cell addition by Togashi. And summarizing, well, you see the population, the total population pressure, and the different these addition forces modeled with the different strengths, using essentially the same potential, okay, but just taking different strengths for the different interactions. So here you have 4 simulations in which here on the side you see the typical strengths that we put between the different type of cells. So red with red, green with green, and green with red. In this case, all the forces are equal. Here you have the different strengths, and then you see this kind of mixing that I mentioned. You just change the different strengths, you get to this case of engulfment, and eventually you get to this kind of partial engulfment, and finally to complete segregation. Okay, so the model was capable to reproduce this cell sorting kind of phenomena, in the sense of reproducing just by changing the strengths of the interactions, that you can go from one type of pattern, the level of the 2 populations of this kind that were described in the biological literature. Okay, sorry about this. Let's see. Okay. So the next thing I want to comment is, of course, we had to be careful in how to match 2 experiments. So we did preliminary work in trying to match to the experimental data by using biologically reasonable parameters. Here you have all described. The only ones that were left for trying to mimic the experiments were the different strengths, and we used the strengths by doing a kind of searching on the different parameters that we had just by playing with the strengths. All the others were fixed from the biological data. So what you have here at the end, this row are the videos of the experiments of Togashi. And here, a one-dimensional simulation of that system of 2 equations that we propose as a way of modeling the the behavior of this population across the 2 populations. Okay, so the first 2 videos corresponds to the first 2 rows of the experiment I showed you at the very beginning of my lecture. Well, the last 2 are all 2 experiments, just to show you that the model is capable to kind of predict some of the behaviors. So as I said, when the cells are not are the same, that they express at the same level the proteins that control the differential adhesion, the adhesion between the different cells, then there is a sharp interface. As soon as you change the strengths, as soon as you have a differential adhesion between the 2 types of cells, then you have a certain intermingling zone, and here you see the area of mixing. And in other cases, you have, like in the last 2 that I will simulate again, you have the case in which one of them somehow invades the other, or an invasion with mixing in this case. And this is just by, as I said, changing the strengths. Okay, so I hope to have convinced you that this model is not only interesting mathematically, but also from the applications in math biology, because it reproduces in several of the experiments that simple experiments for controlling the behavior of cells due to differential adhesion. Okay, let me now go back to the second experiment that triggered my attention, kind of the second phenomena that triggered my attention, which is the pattern formation, the stripe formation in the skin of zebrafish. So let me start by introducing some of the type of cells that are important here, that are responsible for the formation of the patterns on the skin of the zebrafish. It is well known by the groups of biologists working on this for many, many years, that there are mainly three types of cells, so that you can somehow distinguish at least three categories, three families of types of cells that are involved. The xanthophores, that are the ones that lead to the yellowish orange type color in the skin of the zebrafish. The melanophores, that leads to the bluish black color on the skin, and the iridophores, which are the ones that are leading to the silverish color in the skin of a fish in general. So the interaction between these three types of cells, or different type of this type of cells, because you can subdivide them into different type of iridophores, melanophores, and xanthophores, are known to produce this kind of patterns. Okay. And let me see if, yeah, you can see, oh no, I didn't want to show you this, but I want to show you that. Yeah, so maybe you can see this now, I hope. So this is just an experiment by Shigeru Kondo, in which you see a xanthophore, which is the yellowish cell, and the interaction of the yellowish cell with the melanophore. The interesting thing about this is not only that the xanthophore is trying to chase the melanophore, so the yellow cell is trying to chase the black cell, but also the way in which this interaction is produced due to this production of this long protrusion and the formation of certain bonds of the cell. Okay. So, okay. So, in that case, what we started with a very simple model, again of the same type, in which we introduce the total population pressure, the interactions between the xanthophores and the melanophores, which are known to be the ones that trigger the formation of the patterns mainly, and the iridophores that are known to somehow control in the growth of the fish, how they diffuse in the skin of the zebrafish. So we did a very simple model based on these three populations, and we started with that. And let me show you, yes. Let me show you then with that kind of model and adapting the type of potentials, the kind of simulation, I mean, the simulation of the kind of patterns that we see. So just you think about this quadrangle as a very realistic way of looking at the side of the skin of the zebrafish. Is not a growing domain. You could think about doing growing domains too. So here, initially, we just put a distribution of the melanophores, which is more concentrated on the lateral lines and the center, which is known to be idealistically known to be when the fish is born, the distribution of that cells that comes from the primordium. And here, what you see is somehow the formation of the stripes. Led by the diffusion of the iridophores, which is this one. Here is a vertical cut of the simulation here. Here I'm putting melanophores, xanthophores. And what you see is that the model also gives you sharp interfaces, segregation between the areas where the xanthophores are present with respect to where the melanophores are present, and you have these sharp interfaces in between. Okay, so again, this very simple model is able to reproduce the formation of patterns, and here I want to comment that the formation of patterns is not based on any Turing mechanism, it's just based on differential adhesion. Okay, so purely on, mechanical forces. Then we continue trying to improve the modeling, and what we did is first to separate the different type of cells into different phases of the cells that are called dense or loose xanthophores and iridophores. So they are cells in the different type of the same cell, but we were considering as different members of the family, because depending on the phase in which they are, their role in the formation of the pattern of the zebrafish is different. This has been identified in the last 5-6 years by these groups of biologists that we are collaborating with. And here you have all the possible interaction of these type of cells that we were considering and how we were modeling them. All of these interactions between the same type of cells were modeled by volume exclusion, some of them by repulsive, attractive, non-local forces. And then it leads to somehow a much more complicated model in which you have also some reaction terms, because the presence of melanophores or xanthophores were known to produce or to influence the birth and death rates of the other type of cells at a certain distance. So we introduce non-local terms at the reaction level. And by doing so, what we are able is to produce a better fit to what people observed in the formation of the zebrafish, which is that the stripe patterns, they are produced in time from the myoseptum of the fish, so the center, where the cells are really produced. So then without the reaction terms, we will not be able to get this creation of these, the fact that you create there is a growth of another type of cell outside the support, say, of one of the type of cells where one of the cells is present. And well, this is something that we are doing at the moment. And okay, let me go here, and then let me conclude. It's almost time. So, in fact, I didn't discuss that much about the mathematical analysis part of some of the problems here, but let me just mention for those in the audience that are interested about that one of the difficulties here, even in one dimension, for the system of nonlinear diffusion with equations with non-local trees represented at the beginning, is how to get a control on these sharp interfaces. How do you get a control on the BB bounds on the solutions? That's one of the main open questions here. Open question that in case of reaction diffusion, we fixed. We did that in one dimension, and then also in higher dimensions has been done by Benoit Pertam and Piotr Giamsta and Agnieszka Giamsta. And then on the more biological side, I want to just comment that we are trying now at the moment to collaborate with Kondo and Alexandria Folkenin to model these, to get the model for the zebrafish patterning by using essentially input from the obvious the biological experiments, and also from AGM-based models that were derived by Alessandria Folkenin, and we are working on that, also together with my former PhD student, Marcus Smichten, and Shekhar in Sussex. And finally, I just wanted to thank my collaborators in this. Most of the works related to this presentation were done in collaboration with my former PhD student, Marcus Smichten, that is now an associate professor in the University of Dresden. And yeah, with Hideki Murakawa in part, which is in university near Kyoto, in Japan. And thank you very much for your attention.
So thank you, José Antonio, for this very nice talk. And I am very sorry for the technical problems at the very beginning. I had some problem with my connection. I couldn't connect properly. So does anybody has questions? You can type them in the chat, or you can even maybe ask yourself. We can try to unmute you.
We have one question by Maribel Arjona.
Yeah, sure.
So
you just stop my video would be probably better.
Okay. She says that when you talk about engulf, does this mean that one cell is inside of the other?
No, it's around it.
Just around it.
Doesn't mean that one cell is inside the other, just surrounds it. Surrounds it. This is what it means. That surrounds it.
Okay, perfect.
I open also, I have a question or a couple of questions. And then people also have time to write their question.
Sure.
Well, first of all, thank you, José Antonio, for the nice seminar conference. With respect to adhesion,
yes.
Then you distinguish, for example, the cell-to-cell adhesion, that means really local cell-to-cell, or by groups of cells.
Yeah,
that could be a cluster from that you impose, that there is a cluster that remains together, or maybe you put when they move they dimension, for example, the adhesion properties of the cells.
Yeah, well, the thing is that the type of modeling I'm taking here is at the population level. So the density is already, I mean, we are talking about already a density of cells, so it's a macroscopic density. So, I mean, it's for a group of cells already. So what you are trying in this coarse graining procedure is precisely to go from assumptions that you do at the level of the interaction of cells to get a way in which you can write the interaction at the level of populations of cells, which resemble what you assume at the level of individual cell interaction. But yeah, so in these densities of cells are large groups of cells. It's not that you are tracking individual cells, certainly.
And the other question, the other question is related to the kernel. You use as example the absolute value of x, the distance, in fact, between.
Yes, this was just to showcase explicitly some solutions in one dimension.
Yeah, that's a good kernel because it's leeches, you have any property also to the limit, etc. But why this kernel? Yes. There is a biological minimum, or you have tried to modeling some kernel interaction?
Yes. I mean, in fact, as I said, the shape of the kernel is then chosen differently. In fact, one of the papers with Shiana and Colombi, we studied different type of interactions potentials, different choices for the kernel, omega of R. that I use in the models at the end of the day. But yes, I mean the general behavior of the system, of course, changes. The shape of the stationary states, they change with the potential, but the general behavior doesn't. It's quite generic. So I mean, we explored that numerically in 1 dimension. We explored that also in 2 dimensions for the case of the differential addition experiment of Togashi, and there the omega UGR is linear, but cut at a certain distance, so it's completely supported. But it doesn't change that much from the general behavior of the solutions from one potential to the other as soon as they are integrable from the mathematical viewpoint. So, I mean, they can have even some singularity at 0, and things will not change dramatically in terms of the behavior. So, the steady states, I mean.
Okay.
I see some.
Yeah, there are some more questions
popping up in the chat, no?
Yeah, so I'll read them. So in particular, Francesco Cavalieri, he's asking about the mathematical model in slide 27.
Oof.
Which is the meaning for the non-local term?
27, okay. So let me go to the 27. 27. Here. Am I?
We don't see the screen. If you are sharing, we
yeah, sure. But in the 27 there is no. Yeah. Let me. Yes. So this is 28. In 27 there was nothing. So that's why no equation. So maybe it's not this one. But nevertheless, if he's referring to this kind of interactions, is the same as I was discussing in the other model. What you are modeling is the addition or the repulsion forces between different kinds of cells due to an interaction at a kind of average distance. It will be the same. Yes, it will be the same. The non-local model, okay. So probably this slide, will probably clarify better.
Yeah.
Okay, which one here? So the non-local term is modeling the interaction of cells at a certain distance. So the addition forces to the other cells or to the same type of cells nearby. So this part of the interactions I'm modeling at the non-local level.
Okay, and I think there are some more questions, and in particular, there's an interesting question. They are asking why in the simulations for zebrafish you find stripes instead of hexagonal arrays.
Yeah, well, I mean, very good question. We since it's a very complicated model, we didn't study the stability of the stripes. Okay, so I cannot tell you properties of the stability of the stripes. That's a very interesting question that deserves a lot of analysis, also from the numerical viewpoint, to analyze how stable they are, and if you can obtain other type of cells, other type of patterns by mimicking what they have done in experiments. What they have done in experiments is to, for instance, produce mutants by producing zebrafish, in which you artificially kind of kill one of the interactions between these type of cells, essentially by genetically neglecting some of the forces here of attraction and repulsion between some of these cells. And they did that in experiments, and they know that by doing those mutants, they produce different patterns on the skin of the zebrafish. We haven't done that yet with this model. We haven't studied that yet in fully detail, and that's a part of the next steps in this direction. So it's a very interesting question there. Or even in the simplest model here for zebrafish, which is not that realistic, just to understand why the stripes are so stable is an interesting question. Yeah.
Okay, thanks. So I think there are some more actually. And Mauricio is asking, what if, well, I understand, what if instead of looking at an agent base with cell-to-cell interaction, don't you look at a coarse grain model so that there are monomers interacting between each other? So I guess in a sort of small curves, I guess.
We didn't have a look at this intermediate way of modeling. Alessandra Folkeny, one of my collaborators in this part of the zebrafish, she looked essentially to agent-based models. And we have used that experience with the agent-based model to introduce the corresponding terms of the level of the PE. But yeah, I would say an agent-based model makes more sense to me, just because it's not that it's like polymers in which the particles somehow are just beads, and when they produce the bond, they will not, they will just stay like that. So in fact, they are much more active than this. So they can produce the link or the bond or not, and that could change in time. So I'm not sure that one can do something in the middle. So I think it makes more sense to me, coarse graining directly from agent-based models.
Mm-hmm. So, and finally, there is a question by Thomas Hillen, and we can read, I'll read for you. So if you think about cancer, then sometimes cells change their adhesion, detach from the main tumor, and form local metastases. Can you use this model to analyze such a process?
Oh, Thomas, nice question. Thank you. Well, I would say that one could introduce, well, one could introduce that the strength of adhesion depends on several factors, and they change in time for certain reasons. Maybe because of static pressure, maybe because of the level of oxygen, maybe because of the level of blood. So there could be a possibility to include that time variation of the strengths of interaction between the cells. It could be a possibility to think about modeling that kind of behavior. No, we never thought about doing that. Thank you, Thomas.
Yes, it is related. The question of Thomas is related to the question that I tried to pose.
I see. Okay.
Yeah, the same. I don't specify by cancer, but this is in my mind what why in fact when the cell migrate, for example in glioblastoma, usually a group of cell less the real connection, the real adhesion to the others.
And yeah, okay. Okay, no, I mean, of course, you can include in the model this at the level of the strengths, how they change in time with respect to other factors. We didn't explore that. That we didn't.
Then if there is no more question.
Well, I would like to make a comment. José Antonio, I understand that Kondo is the same Kondo that introduced this model for pattern formation in zebrafish, which is Turing-like on growing domains, right?
Right. He's not convinced anymore that Turing mechanism explains it.
That was short of the thing I wanted to ask you.
The same Kondo that participated 2 years ago in BIOMAT.
Yeah, Shigeru Kondo. Yes. No, I mean, he's convinced that the Turing mechanism is not the reason. He has been, he himself has been doing many other experiments in the last 10 years, convincing himself that it's not.
Okay, this is another chapter of the story then.
Yes.
Okay, then thank you again to José Antonio and all the participants in this seminar. I announced that, as you know, that next week Nicola Velomo will speak about some models in COVID, in the pandemic virus. And then thank you very much to all of you.
Thank you for having me here. Anna, I will send you a message, an answer to your question. There was another question that wasn't answered. Don't worry, I'll send her a message.
Okay.
No problem. Okay, thank you very much.
Okay, thank you, everyone.
Bye.
Bye.